Hi everyone. In this lecture, we're going to talk about innate immunity and we're going to talk about the immune system overall. So your immune system protects you against different things that you encounter in your life: viruses, mold spores, cancer, toxins, bacteria, parasitic worms. And here's a picture of a type of white blood cell called a neutrophil, and it's eating mold spores. So phagocytosing is eating mold spores, which is good because you don't want the mold spores in you. So in this lecture, we're going to do an overview of the immune system, and we're going to focus on innate immunity. In innate immunity, there are first line of defenses. We're going to talk about those. We're going to talk about the second line of defenses, and we're going to end the lecture by talking about inflammation and fever. When we look at the immune system is a collection of organs, tissues, cells, typically white blood cells, that are there to protect you against pathogens. And remember, in class we said that pathogens are anything that cause disease. When we look at the immune system, there are two parts to the immune system. So overall, you have an immune system. There's two components. There's innate immunity and adaptive immunity. But just keep in mind that everything is working together in your body rather than just memorizing bits and pieces. And we're going to focus on innate immunity in this lecture, and we'll go back and summarize this slide after I've talked about it. So innate immunity is the part of your immune system that you're born with. So you're innately born with it, and it's a routine protection present at birth. It's what defends you against any pathogen. So sometimes people will say that innate immunity is non-specific. That's what they mean. It's an immediate rapid response to a pathogen, and we have the same typical response in innate immunity regardless of the type of pathogen or the number of exposures. That's innate immunity. Another part of your immune system is adaptive immunity. Adaptive immunity adapts over time and gets better over time. It develops over your lifetime as you're exposed to a lot of pathogens. It has a memory component, so this immune system gets better with time, because if you've been exposed to one strain of influenza, you've built protection in the form of antibodies so that you're able to fight that strain if you get exposed to it again. So that's why we say adaptive immunity gets better with time. This is also the same reasoning why a 20-year-old has a better immune system than a 3-year-old. So a 3-year-old has maybe potentially the same innate immunity as the 20-year-old, but the 20-year-old has had a lot of time to build their adaptive immunity. So again, to summarize, your immune system consists of innate immunity and adaptive immunity, and we're focusing on innate immunity in this lecture. Innate immunity consists of two parts. There is your first line of defenses and your second line of defenses, and this is what we're going to talk about. In your innate immune system, again, this is the part of your immune system that you're born with. Your first line of defenses are the barriers that block pathogens from entering you. So these are the first line of defense that will stop a pathogen from potentially getting in and causing a very bad infection. These consist of your skin, mucous membrane, secretions, and microbiota. So if you can think about it, the first line of protection that we all have is your skin. Your skin is there to protect your organs, your blood, your tissues. And it actually, I'll get back to it when I talk about skin. So if a pathogen does breach or get through your first line of defenses, hopefully your first line of defenses protected you. Your second line of defenses, if your first line of defenses couldn't protect you and the pathogen got in, let's say you have a very bad burn and things got in, your second line of defenses in your immune system will kick in. And these are typically your white blood cells, and we're going to look at different types of white blood cells. And these ones will destroy the invader or the pathogen by doing things such as phagocytosis. So some of your white blood cells are phagocytes. They're cells that can do phagocytosis, meaning they can eat the pathogen or the invader. We have different types of defense proteins, which we'll talk about. There's natural killer cells that help with virally infected cells and cancer cells. And then there's inflammation and fever, which help with limiting different pathogenic infections. So we'll focus right now on the first line of defenses that protect you when you are exposed to a pathogen. And these are your skin, mucous membranes, secretions, and the microbiota. Our first line of defense that protects us is our skin. Our skin, here, let me put this all out. So our skin makes it difficult for microbes or pathogens to get in. And this is the reasoning behind why people who have burns, like third-degree burns, second-degree burns, you keep them in a hospital in a room protected. And typically when nurses and doctors and healthcare workers are going into a badly burnt patient, they're completely covered up because that patient is very immunocompromised and they can easily get infected because they don't have their skin to protect them. The reasons why our skin protects us is, besides being a barrier, it continually flakes off. So your skin cells are always dividing. And by flaking off all the time, you're also removing any attached microbes. Our skin also has keratin, which is a type of protein that repels water. Remember that bacteria and different types of pathogens like moist environments. So if you keep a dry environment, then you won't get infection. And when we look at skin, I know most of you have taken anatomy. It consists of epidermis and dermis. The keratin is in the top layer, the epidermis layer, and there it protects you from getting infected by keeping your skin very dry. And having all these layers and epithelial cells also is another barrier. Another part of your first line innate defenses are your mucous membranes and mucus. So mucus or mucous membranes line the digestive tract, your respiratory tract, and your genitourinary tract. Meaning, if you look at your digestive tract, like your stomach or your intestines or your respiratory tract, this is where you breathe. You have cells that secrete mucus. Mucus is this sticky substance. It's probably the best way I can describe it. You've all experienced mucus in your nose and your throat when you've had a cold or a flu. And the reason why mucus is very helpful in defending you is because it's this sticky substance. It sticks pathogens, and hopefully your body will excrete them before they get even lower. So when you think of a cold or a flu, you don't, or even SARS-CoV-2, which causes COVID-19, the best case scenario is for that virus to get stuck in your mucus. You keep having a bad runny nose and mucus in your throat, and you keep getting that out. So hopefully that virus does not get into your lungs. And that's the purpose of mucus. So it traps dust and pathogens, and there's something called the mucociliary escalator in our respiratory tract. So when you look at our respiratory cells, these are cells that you use to breathe. Those cells have cilia on them, which are little projections, and they have cilia and mucus. What happens is the mucus will trap the pathogen and the cilia will sweep it up so that you don't get the pathogen to go deeper in your lower respiratory tract, your lungs. So this is where this becomes very helpful. Another first line of innate defenses is different secretions that our body makes. So these secretions, let me put this all out. These secretions are antimicrobial. They have different antimicrobial properties, enzymes. When we look at secretions like tears, saliva, oils, sweat, they have different chemicals in there that inhibit pathogens from growing. So for example, when we look at tears and saliva, they have the enzyme called lysozyme. Lysozyme degrades peptidoglycan. And remember, we saw peptidoglycan in the cell walls of bacteria. And also when we look at sweat, for example, sweat is very salty. And remember, we learned that most bacteria don't like growing in hypertonic environment. Your stomach is very acidic. This also inhibits a lot of organisms because most organisms are neutrophils and are killed by acids. And then saliva, tears, and urine also play a role as flushing actions. So when you cry, you get rid of any potential infection you could have had in your eyes. Saliva also is always constantly getting rid of stuff. Urine, a lot of times when people have UTIs, we tell them to drink a lot of water so they can urinate. And it's a self-limiting infection where a lot of times they don't need anything. If they can drink enough water and flush out the microbe, they're good to go. And then finally, a first line innate defense is your microbiota. Your microbiota are the good organisms that you're born with. So the good bacteria that we see that you're born with, and it's on different parts of your body. So they're good microbes in many parts of the body. And the reason why they protect you is if you have good bacteria and they're covering your body, bad bacteria or bad microbes, pathogens, don't have space to grow and they can't get nutrients. So that's how they're protecting you. They prevent pathogens by covering different binding sites on your body by using nutrients. This is also one, okay, so there's been a lot of research studies saying that babies who are born by vaginal delivery, so natural vaginal delivery, have a stronger initial immune system than babies born by C-section. Babies born by C-section is when you cut the mom's stomach open and you take the baby out. The reasoning behind this is when we look at the vaginal canal, there's a lot of really good microbes there. And if the baby gets to pass through that, it develops a potentially stronger microbiota. Whereas when we look at babies who are born in somewhat a sterile environment where you just cut the mom's stomach open and you take up the baby, they do not develop as strong of a microbiota initially. Over time, things change. So that's why some researchers have studied that if you take babies that are born by C-section and you take some of the mom's vaginal fluid and you rub it on the baby right when they're born, maybe they'll have a better or stronger microbiota to protect them. And the reasoning is that these microbes are covering binding sites where potential pathogens would have maybe grown or infected. So we talked about the first line defenses. Now we're going to talk about the second line of defenses. What happens when all of these things couldn't protect you and the microbe actually got in? This is where your white blood cells kick in, inflammation, and fever. And this is what we're going to talk about now. So when something has gotten deep through your skin, your mucous membranes couldn't get rid of it, whatever, it got through. This is where your white blood cells are going to play a part. And before we talk about white blood cells, I wanted to talk about the formation of blood in general. Just one quick slide. Put this up. So blood cell formation is called hematopoiesis, and blood cells originate from hematopoietic stem cells, which are found in bone marrow. Bone marrow is if you take a bone and you split it in half. When you look inside, there's kind of like this substance in the middle, that's the bone marrow, and you see their hematopoietic stem cells. This is where blood cells originate. When you look at blood, there's three components to blood. There's your red blood cells, platelets, and white blood cells. The function of red blood cells is to carry oxygen in the blood. Throughout your body, they carry oxygen, and we call red blood cells erythrocytes. Remember, cyte just means cell. Platelets are important for blood clotting, and white blood cells, which are called leukocytes, are important in your immune system, which is what we're going to focus on. We're going to focus on leukocytes, but I want everyone to know that blood has three components. So red blood cells, which we call erythrocytes, platelets, which are important for clotting, and white blood cells, which we call leukocytes. There's also the fluid component, which is the plasma part of blood. Leukocytes are just the fancier term for white blood cells. They mean the same thing. We have different types of leukocytes or white blood cells in our blood. So and they're named sometimes based on what they look like. Let me put that all out there. One type or one category of leukocytes or white blood cells are granulocytes. We call these types of cells granulocytes because when you look at the cells under the microscope, they look granular. They look like there's little grains inside them. And I'll show you pictures of them in the lab slide, which I'll record later. So with the granulocytes, neutrophils are granulocytes. This is what neutrophils look like. And neutrophils are important for phagocytosis, for eating, or for phagocytosing different pathogens. Basophils and mast cells. So here's a basophil. It looks granular. They're important for inflammation. So they have histamine inside. Eosinophils. This is what eosinophils look like. They're important if you get a parasitic worm infection, and they're also important in inflammation. Cells that are not granulocytes or agranulocytes are monocytes and lymphocytes. Monocytes are macrophages and dendritic cells. Macrophages are very important in phagocytosis, and so are dendritic cells. Dendritic cells will be important in adaptive immunity, which we'll learn about later. And then lymphocytes are natural killer cells, and T cells and B cells. Natural killer cells are important for helping fight off anytime you have a virus or cancer/tumor cells. T cells and B cells are a type of lymphocyte, and we'll learn about them when we talk about adaptive immunity. So I want you to know the different functions of these cells, and when we do the lab video, I'll show you what they look like this under the microscope. And I want you to keep in mind what was the function of each cell. I highlighted macrophages and neutrophils because they're very important cells in your immune system because they do phagocytosis. Phagocytosis is very important part of protecting you against different infections. Phagocytosis is when cells engulf, basically eat and digest pathogens. They just chop it up. And phagocytosis are done by phagocytes, or cells that can do phagocytosis. The white blood cells that do phagocytosis are macrophages, neutrophils, and dendritic cells. How your body knows what cells to phagocytose and what not. So I'm going to try to make this as brief as possible. A lot of pathogens look different than your cells. So when a pathogen is ingested or taken in, it looks different because they have different patterns than your own cells. And we call these different patterns pathogen-associated molecular patterns. So an example of something like a PAMP is peptidoglycan. Your cells do not have peptidoglycan. So when you swallow something, a pathogen that has a cell wall made out of peptidoglycan, your cell says, this is foreign. I need to tag it for some sort of destruction, such as phagocytosis. The part of your own cells that recognizes these patterns are called pattern recognition receptors. So on your white blood cells, you have different receptors, and these receptors are what are going to go and recognize the different molecular patterns that are foreign on the pathogen. And the receptor from your cell will bind the different PAMPs, pattern-associated molecular patterns, and then this will trigger an immune response. And one type of pattern recognition receptors are toll-like receptors. I put this out there in case you are reading the textbook or you see this again in the future. There's a lot of studies that are being done on toll-like receptors. They're the receptors on your own cells that will recognize foreign components on different pathogens. So pathogens have unique components, and your host cell receptors attach to the PAMPs and release signaling molecules. Signaling molecules are called cytokines, and these cytokines will go and trigger some sort of immune response, such as phagocytosis. So this is a picture of phagocytosis. Here's a microbe. And the toll-like receptors, or the pattern recognition receptors on your own white blood cells, were able to adhere to a different PAMP on this pathogen and then trigger phagocytosis. And again, you have cytokines released, which are signaling molecules. So we're going to talk about the process of phagocytosis here. I'm going to go over this picture and then show you guys the notes. Your white blood cell, the phagocyte, such as the macrophage or the neutrophil, they will recognize a microbe and they will go and adhere to it. So they're going to go and stick. And remember, the microbe has the pattern associated molecular pattern, and your cells have receptors, such as toll-like receptors. They'll adhere to that microbe. And once they adhere to it, they'll ingest it. So that phagocyte, that white blood cell, that neutrophil, that macrophage will ingest it. And now the microbe is inside your white blood cell. Now your white blood cell will form a phagosome, which is just a vesicle. And that vesicle will fuse with a lysosome. If you guys remember when we talked about cells, lysosomes are vesicles that have a lot of digestive enzymes. So when the phagosome with the microbe fuses with the lysosome, you have the phagolysosome, and all the enzymes in the lysosome will go and break or kill the microbe. There's a lot of toxic oxygen radicals, lots of toxic material here. They'll digest the microbe, and then the microbe will get released from the cell after it's been digested. So this was phagocytosis. And phagocytosis is done by neutrophils, macrophages, and dendritic cells, which are all leukocytes, meaning they're white blood cells. So this is just basically what I said, and then you kick out the remains or the waste remains of the microbe. Now, phagocytosis steps are important here, all the way from recognition to getting it inside, to putting it in a phagosome, to getting the lysosome to fuse, to release. These are all very important steps because a lot of pathogens, the reason that they've been so good at infection is because in one way or another, they've evaded different parts of these steps. So for example, the bacteria that causes tuberculosis, a very bad lung infection, has come up with effective ways to live in a macrophage, which causes phagocytosis, and not be harmed in this process. So scientists study this process to see why have bacteria been able to infect. If all your cells can do good phagocytosis, get rid of the microbe, why do they still cause bad infection? And it's because microbes have come up with clever ways to maybe not be recognized. Maybe once they're recognized, they can live in the phagosome. Maybe they somehow stop the lysosome from fusing with the phagosome. So different components. So we talked about cells that do phagocytosis. You have a lot of other really important white blood cells that do other things. So basophils and mast cells are important in inflammation, which we'll talk about in the end. Natural killer cells are also a type of white blood cells that helps destroy virally infected cells and tumor cells or cancer cells. And then eosinophils, you see them when someone has a worm infection or parasitic infection, the number of eosinophils goes up. They also help with inflammation. I want to have one slide about cytokines to talk about cytokines really quickly because we'll see this word a lot in this lecture, and then adaptive immunity. Cytokines are signaling molecules. They're small proteins that are secreted by cells, and they are signaling molecules, meaning when a cell secretes them, they signal another cell to do something. So they affect the behavior of other cells. They're secreted by different white blood cells, and they're secreted by neutrophils when they encounter a pathogen. So when a neutrophil sees a pathogen, it secretes cytokines, tells your body that we have an infection going on, we need to fight it. Same thing with macrophages. They secrete different signaling molecules when they see pathogens. Natural killer cells, when they encounter a virus-infected cell or cancer cell, they also release cytokine signaling molecules. And lymphocytes, which will play a role in adaptive immunity in the next lecture, also secrete cytokines. And again, cytokines will go and signal your body or your cells to do something. They're basically your white blood cell is saying, we saw a pathogen, let's secrete some molecule called a cytokine that will tell your body we have an infection. Let's maybe do inflammation. Let's turn on a fever, something like that. Now we're going to talk about another second line innate defenses, which are defense protein substances. When you look at your body, you have different types of proteins that defend you against infection, and we lump them into defensive protein substances. So the different defense protein substances that we're going to talk about antimicrobial peptides, interferons, and the complement system, which are all made up of protein system. So the first one, antimicrobial peptide system. Antimicrobial peptides are proteins that protect you against bacterial infections, and they're made by your skin, different mucous membranes. Remember, we said that your digestive tract, respiratory tract, and genitourinary tract make mucus, and blood cells, they all make these little short proteins called antimicrobial peptides. And what they do is antimicrobial peptides go and coat bacteria and destroy it by creating holes in the bacteria and stopping the bacteria from being able to form its cell wall. So they're produced in response to protein and sugar on microbes. Your body, again, recognizes that there's a foreign invader. And these antimicrobial peptides, these little small proteins, will inhibit cell wall synthesis, and they'll form pores or holes in the plasma membrane of the microbe. They have a broad spectrum of activity, meaning antimicrobial peptides target a lot of different types of bacteria, not just one. And the outcome is that they kill the foreign invader and they stimulate an immune response. Stimulate an immune response means maybe you stimulate fever or inflammation to happen. Another type of defense proteins are interferons. Interferons are, let me put this out. So interferons are cytokines. They're a type of signaling molecule. They're a signaling protein, and they're made by virally infected cells. They cause nearby cells to go through cell death, which we call apoptosis, if they become infected with a virus. So they're very important antiviral proteins that are secreted when a cell is infected with a virus. Remember, with virus infection, you have a virus, it infects a cell, and then it uses the cell's properties to make a lot more viruses, and then those viruses go out and infect a lot of cells. So if you can stop the viruses from infecting a lot of other cells, you can limit the viral infection so that your immune system can fight it off. And this is how your immune system fights it off. So interferons, the name comes from they're proteins that interfere with virus replication. So what happens is you have a virus that infects a cell, maybe, for example, SARS-CoV-2, which causes COVID-19. You have this virus, it infects the cell. Remember, with the virus, it causes your cells to, it uses your cell's material to make more of itself. Anyway, the infected cell, your infected cell, will release interferons, which are cytokines or signaling molecules. And these interferons will go and warn cells to stop virus replication. And it's a very efficient way of stopping a viral replication from continuing. So interferons will go and contact other uninfected neighboring cells. And when they contact them, they basically tell them, stop the viral replication cycle. It's a lot more complicated than that. I just wanted you guys to have a simple idea of what happens. And just so you guys know, if we did not have interferons, many of us would die from potentially simple infections such as influenza infections. And the outcome is you prevent viral replication in infected cells. If you stop virus replication, you basically stop the infection. Another defense protein substance is the complement system. The complement system is a set of protein, and its name comes that it complements activities of your immune system. So it's a series of proteins that we see circulating in blood and tissue fluids, and there's complement 1 to 9 proteins, so they're labeled C1, C2, C3, C4. And they're named in the order they were discovered, C1 through C9. C stands for complement. They can split into fragments. So for example, C3 protein splits into C3A and C3B. By the way, the complement system can be a whole lecture on its own, so I'm just trying to give a brief slide of what it does. Complement proteins, what they do is they bind to microbes. So you have a pathogen. What they'll do is let's say it's a bacteria, they'll come and they'll bind to it and they'll coat the microbe, and they'll lead to its destruction through one of 3 ways. So again, they'll bind the microbe. They can lead to phagocytosis, which we call opsonization. So opsonization is a way that we facilitate phagocytosis. They can directly lyse the microbe by forming these complexes, which we call MAC complexes, or they can lead to inflammation, which also leads to the death of the microbe. So this is what the complement system can do. And you get various triggers. It's a very long, complicated process. I just want you to know these things. And once you have complement coating a microbe, you either get lysis of the foreign cells because the different proteins, the complement proteins, create membrane attack complexes, which cause the cell to lyse. This is what we call cytolysis. You can lead to the microbe being phagocytosed, which is what we call opsonization, or this coating of complement proteins on the microbe leads to inflammation. And we're going to talk about why inflammation also kills the microbe. So all of these things are great things that complement proteins do. You either get lysis of the microbe, which is good. You get phagocytosis of the microbe, or you get inflammation, which also it ends up limiting or killing the microbe. Now we're going to talk about inflammation. Before I mention inflammation, I want to say that many different cell types can lead to inflammation, and inflammation happens when cells are infected or damaged. So this is a microbiology class, so we're talking about infection with different microbes, but as you know, you can get inflammation when cells are damaged. So if you hit your hand really hard, you can get inflammation there because those tissues are damaged. And the purpose of inflammation is to contain the site of damage or infection, to localize the response, to eliminate the invader, the pathogen, and to restore the function of the tissue. Inflammation results in swelling, redness, heat, pain, sometimes loss of function. Sometimes inflammation can be so bad that it damages tissue. All different types of cells a lot of times will lead to inflammation. And I'm talking about white blood cells. So neutrophils will lead to inflammation, monocytes, macrophages, natural killer cells, signaling molecules, cytokines, interferon. The complement system, one of the things that leads to is inflammation. Inflammation is good because it limits infection. Too much inflammation can be bad. We'll talk about that later. So with inflammation, you have different chemicals that are released so that you can get inflammation going. These are called pro-inflammatory mediators. So when cells are damaged or infected, you get these pro-inflammatory mediators released. And these chemicals that are released to say that we need to get inflammation going are histamine, prostaglandins, and leukotrienes. And this is why you see a lot of times a lot of antihistamine drugs. So when people have too much inflammation, we see antihistamine drugs. We're going to learn about it during allergies later on. Process of inflammation is you have tissue that's injured. Let's say infection. Let's not say injured because it's a microbiology class. So you have tissue that's infected. Maybe you have something that goes in your hand that had bacteria in it, or a virus gets in. So what happens is you get inflammation going. Vasodilation happens. Vaso means vessel. Dilation means get big. And so here's a normal blood vessel. Inflammation leads to vasodilation. Because your vessels, your blood vessels are bigger, you get increased permeability. And so you get more phagocytes, which are good, different types of good white blood cells to go in and phagocytose the microbe that got in. So you cut yourself, a microbe got in, your blood vessels will dilate so that fluid can get in, fluid that has white blood cells. Some white blood cells are phagocytes. They'll go on and they'll go to phagocytose or eat the microbe. And then the pus is basically all the white blood cells that have killed the microbe, and then you get clearing of debris. Remember, macrophages are a type of white blood cells. So when we look at these pictures, you see that when you have vasodilation, when your blood vessels dilate, you get increased permeability, which is a good thing because you want your white blood cells to get in to be able to get rid of the microbe or pathogen. And the specific process of how inflammation goes, you get tissue injured. The injured cells or infected will release chemical signals. Remember, different pro-inflammatory mediators are histamine, prostaglandins, leukotrienes. So chemical signals such as histamine will cause nearby blood vessels to dilate. So you get a lot of fluid leakage. And when you have a lot of fluid, you can dilute toxins and microbes and bring extra oxygen, which will help with healing. Fluids contain also the complement proteins, which help, remember, get rid of the microbe by 3 different ways. By opsonization, which is phagocytosis, you can cause the microbe to lyse, or inflammation. So this is the inflammation part. Then you get your white blood cells, your leukocytes, to get to the infected tissue. And the fluid in the vasodilation will also deliver clotting factors that will help just kind of put a wall on the site of infection and stop the spread of microbe, prevent bleeding. the neutrophils that came in, so the type of leukocyte, the neutrophil, will go in and do phagocytosis. The tissue debris, the dead neutrophils, will accumulate as pus. That's what pus is, that little white that you see, the little white fluid that you see after an infection is dead white blood cells, neutrophils, and different leftovers from your tissue. The outcome of inflammation is you destroy the agent, so you destroy the microbe that potentially could have gone on to cause a very bad infection. You localize the response to an area, so you localize the response maybe to your hand, and all of this will eventually restore function to that damaged tissue. So I put a question here. Would these same responses be beneficial if they were chronic or system-wide? So chronic inflammation is when you always have inflammation. Is this a good thing? Do we want our blood vessels to always be dilated? Do we always want macrophages there and neutrophils? And if inflammation was system-wide, it wasn't limited to your hand, it was all in your body, is this a good thing? And as you can imagine, the answer to these things is no. We don't want chronic inflammation, and system-wide inflammation is not a good thing. With inflammation, not everything about inflammation is good. I do want to say, though, a little bit of inflammation is good. It protects us, but too much inflammation, chronic inflammation, can lead to tissue damage and cancer. So we're going to do a brief explanation why. Acute inflammation is short-term inflammation. This is a good type of inflammation, mainly done by neutrophils, your white blood cells that do phagocytosis, and macrophages clean up the damage by ingesting dead cells and debris, the different waste material. If acute inflammation fails, you can get chronic inflammation. Chronic inflammation is inflammation that's always happening, and you get a lot of macrophages accumulating. This is not a good thing because macrophages hold a lot of toxic components that are there to kill pathogens. So too much accumulation of macrophages, you'll get these granulomas forming. And doctors that look at tissues, when they look at, for example, stomach tissue, inflamed stomach tissue versus regular stomach tissue, when you look at chronic inflammation, you see a lot of these granulomas. And it leads to tissue damage. Again, the tissue can eventually die over time because of all the accumulation of macrophages. Macrophages are there to kill pathogens, but too much of them in tissues can release toxins that are in the macrophages. One type of toxin in there that's supposed to protect you against pathogen are reactive oxygen species. Too much of this can be bad and cause mutations that can lead to cancer. You can also injure tissues. You can permanently damage tissue. That's why too much inflammation is not a good thing. And people typically take steroids, cortisone to manage inflammation. But a downside of taking these drugs is you can see that people who take too much cortisone and steroids will tell you that they may potentially have a dampened immune system because of these drugs, because inflammation also has a good aspect to it. Finally, the last slide we're going to go over here is fever. Fever is part of your innate immune system. So fever is abnormally high body temperature. Normal body temperature is 37 degrees Celsius, and it's regulated by your brain, a specific area in your brain called the hypothalamus. Anything over 37.8 degrees Celsius, we consider a fever, and fever always indicates an infection. So your brain regulates your temperature, the hypothalamus specifically, and this temperature is raised during an infection, and it's raised in response to things called pyrogens. They're chemicals that your body makes in response to microbes or microbes themselves. So this is how fevers happen. You get an infection, and cytokines are released by macrophages, the white blood cells, causing your brain to release prostaglandins. The specific signaling molecule that's released that tells your brain to release prostaglandins is pyrogens. So remember, cytokine just means signaling molecule. So you get an infection. Pyrogens are released by macrophages. So the macrophages are the ones that ingested the microbe. Now the macrophages are releasing the cytokine, pyrogens, telling your body we need to raise the temperature. And The hypothalamus releases prostaglandins. Prostaglandins reset the hypothalamus to a higher temperature. And the reason why this is a good thing to fight off infection is because remember how microbes have an optimum temperature they like to grow in? So if you raise the temperature, they don't like growing in that temperature anymore. And you also start potentially denaturing their enzymes. Another reason why raising the temperature to fight an infection is good is because, as you guys remember from chemistry, higher temperature meaning that reactions happen faster, so your enzymes go on and do reactions faster to help you get rid of the infection. And one reaction to fever is shivering, chills. So if someone, the reason I put this here is if someone doesn't remember if they had a fever in the night, a lot of times the doctor will ask them, did you have shivering or chills? And these are reactions to fever. So they indicate that a fever had happened. The outcomes of a fever is you get the microbe inhibited, again, because microbes don't like temperatures that are not their optimum temperatures. The optimum temperature for most microbes to grow is 37 degrees Celsius. So when you raise that, the microbe isn't happy anymore. And then your body defenses are accelerated so that they can fight off the infection. And all of this is thanks to the pyrogens that are released by the macrophages that had encountered the pathogen. So this response is systemic, meaning it's system-wide. It's not like inflammation. Inflammation, you can have like inflammation on your hand, inflammation in different parts of your body. Fever is all throughout your body. So we say that it's systemic. Should all fevers be treated? This is a very important question in medicine that people deal with all the time. If a child has a fever or an adult has a fever, should you let them have a fever? Should you treat it right away? And the thinking right now is a short-term fever, of course, you need to check situation by situation, should be left on its own because it's your body's way of defending itself against different pathogens. But if a fever becomes long-term, then you should treat the fever because it can start harming things in your body. But initially, fevers are good. And anti-inflammatory drugs and drugs that reduce fever try to dampen the normal inflammatory response and help reduce swelling and fever. So just different drugs that people take to reduce inflammation, reduce fever. So if we look at aspirin, it reduces pain. So aspirin, Tylenol, acetaminophen is a chemical in Tylenol, and ibuprofen, they reduce pain, reduce fever. But when we look at aspirin, it reduces inflammation, so which we do not see in Tylenol as much. So a lot of times, a lot of people are only supposed to take Tylenol for certain infections. People who have chronic inflammation and different types of disorders learn about it with immune disorders, who have inflammation all the time, sometimes are advised to avoid any drugs that are similar to aspirin because of this. And finally, everything we learned about so far was called the innate immune system. We learned about the first line defenses, the second line defensive, and then the next lecture will be adaptive immunity. So this is it for this lecture, and I hope you guys can slowly take the parts together and keep in mind that all of these are happening at the same time. So with that, I'll do the adaptive immunity lecture later.